Hi everyone, thank you so much for being in this video. We're going to talk about the sense of taste. My name is Eslin. Let's begin. Taste is characterized as a chemical sensation in which various tastings containing food or beverages interact with taste receptors. There are 5 basic tastes, which are salty, sour, bitter, sweet, and umami. The dorsal surface of the tongue has mucosal irregularities and elevations called lingual papillae. We have filiform papillae. They are small and they are the most numerous and only serve a mechanical role. We have also foliate papillae. They have a parallel organization and they help identify sour and bitter. We also have fungiform papillae. They are mushroom shaped and they identify salty and sweet. And we have circumvallate papillae. They are large, dome shaped, and identify sour and bitter. Inside the lingual papillae, we have the taste buds. They perceive taste sensation through sensory cells. Here we have a taste bud, and it contains neuroepithelial cells, which are the sensory ones, and also supporting cells, as well as basal cells. The turnover time of these cells is about 10 days. And they can be found in other places besides the tongue, like the glossopalatine arch, the soft plate palate, the posterior surface of the epiglottis, and the posterior wall of the pharynx. Tastes. First we have salty. Salty is associated with salt, like the sodium ion specifically. Here we have a neuroepithelial cell, and we have in green sodium ions. When we eat something salty, the sodium ions goes through the amylorate sensitive sodium channel. That causes depolarization of the cell membrane and causes or make voltage sensitive sodium and voltage gated calcium channel to open. They cause that depolarization cause these channels to be open. Then the calcium that comes through the voltage gated calcium channel stimulate the synaptic vesicles that then release the neurotransmitter, and later on will be interpreted for our brain as salty. Here we have sour is associated to acid food and associated mainly with hydrogen ion. Here we have a neuroepithelial cell, and we have here in purple the hydrogen ions. When we eat something acid, the hydrogen proton blocks the potassium channel, and then the hydrogen enters to the cells through the taste specific hydrogen channels and the amylorate sensitive sodium channel. That causes depolarization of the cell membrane, which activate the voltage gated calcium channel that allows the calcium to go through it. That triggers migration of the synaptic vesicles, which activates the neuron, and later on in our brain is interpreted as sour. Finally, we have bitter, sweet, and umami. These tastes are G protein coupled. And we have in blue the bitter receptor, in yellow we have the sweet receptor, and in green we have the umami receptor. When the tasting substances get in contact with the receptor, they activate the G protein coupled to them, which activates the phospholipase C enzyme. That causes the IP2 to be converted to IP3. The IP3 opens sodium channels and also activates the calcium store, the internal calcium store. As the sodium ions pass through the sodium channel, that causes depolarization of the cell membrane that activates the calcium channels that are sensitive to voltage, and that causes the calcium to get in the cell. The influx of calcium from outside the cell and from the internal cellular store triggers migration of the synaptic vesicles that causes depolarization of the neuron, and later on our brain interprets that as bitter, sweet, or umami. It would depend of the receptor that is activated. Neurophysiology. The special innervation of the tongue is divided in two parts, anterior part and posterior part. The anterior 2/3, which is the pink area, is innervated by, the innervation is supplied by the chorda tympani branch of the facial nerve, and the posterior third, the purple part, the posterior part, is supplied by the lingual branch of the glossopharyngeal nerve. The anterior part, which is pink, the chorda tympani of facial nerve, and the posterior part it's supplied by the lingual branch of the glossopharyngeal nerve. Let's talk a bit about the taste pathway. The facial nerve and glossopharyngeal nerve goes to the nucleus of the solitary tract in the brainstem. This is the first neuron. The second neuron goes to the ventral posterior medial nucleus of the thalamus. This is the second neuron. And the third neuron goes to the gustatory cortex in the parietal lobe. The gustatory cortex is the Birdman area 43. And that's the taste pathway. And thank you so much for watching this video. As you might noticed, I'm not a native American speaker. I just learning English, and I love medicine, and I'm trying to practice. And I had this presentation in Spanish, so I said it would be good for me to translate it and present it. So I hope you could understand, and I hope that this video was helpful to you. Again, thank you so much for watching this video. If you like this content, please subscribe to my channel, Mediversia, where we have mainly Spanish content, but from now on we will try to introduce English content as well. See you next time.